Hello and welcome back to PharmaHealth Insights, and you are watching our Bioequivalence Studies Masterclass. In the previous episodes, we started from the very basics. We first understood what bioequivalence actually means. Then we followed the journey of a drug inside the human body and understood bioavailability. And in episode 3, we discussed a very important practical question. Which pharmaceutical products actually require bioequivalence studies? Now, after watching that episode, a very natural question may come to your mind. If a generic pharmaceutical company wants to demonstrate bioequivalence, does it always have to conduct a clinical bioequivalence study in healthy volunteers? Does every generic tablet need to be administered to human subjects? Does blood always have to be collected at multiple time points? Do we always need to measure plasma drug concentrations and calculate parameters such as Cmax and AUC? The answer is no. In certain scientifically justified situations, a pharmaceutical company may be allowed to avoid an in vivo bioequivalence study, and this is where the concept of a biowaiver comes into the picture. But please understand one thing very clearly. A biowaiver does not mean that the regulator is simply ignoring bioequivalence, and it definitely does not mean that the manufacturer can launch a generic product without sufficient scientific evidence. A biowaiver means that, under specific conditions, bioequivalence may be demonstrated using scientific evidence other than an in vivo human bioequivalence study. And that is exactly what we are going to understand in today's episode. What is a biowaiver? Why are biowaivers allowed? What is the Biopharmaceutics Classification System, or BCS? What are BCS Class I, Class II, Class III, and Class IV drugs? Which drug products may qualify for a BCS-based biowaiver? What is the role of solubility, permeability, and dissolution? And most importantly, why can two tablets showing similar dissolution sometimes avoid a human B study, while two other tablets with apparently similar dissolution cannot? By the end of this episode, you should have a clear conceptual understanding of biowaivers. So stay with me until the end. Let us begin. Let us start with the word itself, biowaiver. In simple language, it means that the requirement for an in vivo bioavailability or bioequivalence study may be waived. Normally, when we compare a generic tablet with a reference product, we may conduct an in vivo bioequivalence study. Healthy volunteers receive the test product and the reference product. Blood samples are collected, the concentration of the drug in plasma is measured, and pharmacokinetic parameters such as Cmax and AUC are compared. But in a biowaiver approach, we may not need to conduct this human pharmacokinetic study. Instead, under defined scientific and regulatory conditions, laboratory-based evidence may be used to support equivalence. One of the most important tools used for this purpose is the in vitro dissolution study. Now, some of you may immediately think, Sir, are you saying that a dissolution test can replace a bioequivalence study? The correct answer is sometimes, but not for every drug and not for every formulation. This is extremely important. A dissolution test and a human bioequivalence study are not automatically interchangeable. For a dissolution-based approach to be scientifically acceptable, we must first understand the properties of the drug substance. And this brings us to one of the most important concepts in biopharmaceutics, the Biopharmaceutics Classification System, commonly known as the BCS. Before understanding the four BCS classes, let us first understand the scientific problem. Imagine that you swallow a tablet. For the drug to reach the systemic circulation, two major things generally need to happen. First, the drug must become available in solution. In simple words, the drug must dissolve. Second, after dissolving, the drug must cross the intestinal membrane and enter the body. This is called permeability. So, from a simplified biopharmaceutical perspective, two important properties are solubility and permeability. Think of it this way. A drug molecule is trying to enter a building. First, it must be available at the entrance. That is similar to dissolution and solubility. Then, it must be able to pass through the door. That is similar to permeability. If the drug does not dissolve properly, there may not be enough dissolved drug available for absorption. And even if the drug dissolves very well, if it cannot cross the intestinal membrane efficiently, absorption may still be limited. Based mainly on these two properties, drug substances can be divided into four BCS classes: BCS Class I, BCS Class II, BCS Class III, and BCS Class IV. Let us understand each class carefully. BCS Class I, high solubility, high permeability. BCS Class Y drugs have high solubility and high permeability. This is the most straightforward category from a biopharmaceutical perspective. The drug dissolves well, and once dissolved, it crosses the intestinal membrane efficiently. So both major steps are favorable. Let us use our building example again. The drug is available at the entrance and the door is wide open. Because of these characteristics, the absorption of a class 1 drug is generally less likely to be limited by poor solubility or poor permeability. Now think about a generic immediate release tablet containing a BCS class 1 drug. If the generic tablet and the reference tablet both release the drug rapidly, and if their dissolution behavior is sufficiently similar, then scientifically, we may be able to predict that their in vivo performance will also be comparable. This is one of the major scientific foundations of the BCS-based Biowaiver concept. For eligible immediate release products containing suitable BCS Class I drug substances, an in vivo B study may sometimes be waived if all applicable regulatory conditions are satisfied. Please notice my words carefully. I am saying may be waived. I am not saying that every BCS Class I drug automatically receives a biowaiver. There are additional conditions. We will discuss them shortly. BCS Class II, low solubility, high permeability. Now let us move to BCS Class II. These drugs have low solubility but high permeability. This means the drug can cross the intestinal membrane efficiently, but the problem is getting enough of the drug into solution. The door is open, but the drug is struggling to reach the door in a dissolved form. For Class II drugs, dissolution can become a very important factor controlling absorption. Let us imagine two tablets containing the same Class II drug. Tablet A releases and dissolves the drug relatively quickly. Tablet B releases the drug much more slowly. Because the drug already has poor solubility, this difference in dissolution may significantly influence the amount and rate of drug available for absorption. Therefore, formulation differences can have a major impact on in vivo performance. Particle size may matter. The crystalline form of the drug may matter. The manufacturing process may matter. The type and quantity of excipients may matter. Even factors affecting wetting and disintegration may become important. This is why BCS Class II products generally require much more careful evaluation. A simple statement that both tablets contain 500 milligrams of the same drug is clearly not sufficient. The drug may have excellent permeability, but if its dissolution behavior changes, its bioavailability may also change. This is one reason why biowaiver approaches are not universally applicable to all drug substances. BCS Class III, high solubility, low permeability. Now let us look at BCS Class III. These drugs have high solubility but low permeability, so the drug dissolves easily. But crossing the intestinal membrane is the challenge. In our building example, the drug reaches the entrance without any problem, but the door is narrow. Because permeability is low, absorption may be sensitive to factors that influence the interaction of the drug with the gastrointestinal environment and intestinal membrane. Now, this creates an interesting situation. If the drug dissolves very rapidly, dissolution itself may not be the major limiting step. However, the formulation still needs careful attention. Why? Because certain excipients may potentially influence gastrointestinal transit, intestinal permeability, or other factors related to absorption. Therefore, for BCS Class III drug products, regulatory expectations regarding formulation similarity can be particularly important. In many biowaiver frameworks, the excipient composition of the test product is expected to be carefully controlled and appropriately similar to the reference product. The exact regulatory expectations depend on the applicable guideline and market. So, Class III products may qualify for a BCS-based biowaiver in certain situations, but the formulation and excipient assessment becomes extremely important. BCS Class IV, low solubility, low permeability. And finally, we have BCS Class IV, low solubility and low permeability. Here we have challenges at both stages. The drug does not dissolve easily, and even after dissolving, it does not cross the intestinal membrane efficiently. The drug is struggling to reach the door, and the door itself is difficult to cross. From a biopharmaceutical perspective, these drug substances can be particularly challenging. Formulation development may be complex, absorption may be variable. Predicting in vivo behavior purely from routine dissolution testing can be difficult. Therefore, BCS Class IV drugs are generally not considered straightforward candidates for BCS-based biowaivers. So if you want to remember the four BCS classes in the simplest possible way, remember this. Class 1, high solubility, high permeability. Class 2, low solubility, high permeability. Class 3, high solubility, low permeability. Class 4, low solubility, low permeability. But please do not simply memorize this table. Understand the logic. Solubility tells us whether the drug can become available in solution. Permeability tells us whether the drug can efficiently cross the biological membrane, and the combination of these properties helps us understand the possible rate-limiting steps in drug absorption. So, which BCS classes may qualify for a biowaiver? Now we come to the obvious question. Which BCS classes are considered for BCS-based biowaivers? Traditionally, BCS Class I drugs have been the most widely recognized candidates. High solubility, high permeability, and when formulated as suitable immediate release products with appropriate dissolution characteristics, their in vivo behavior may be sufficiently predictable. BCS Class III drug products may also be considered under certain regulatory frameworks, subject to more stringent conditions, particularly related to rapid dissolution and excipient composition. But remember, the regulatory approach may differ depending on the country, agency, and applicable guideline. For example, expectations should always be checked against the current requirements of the relevant regulatory authority. Therefore, never prepare a regulatory submission simply by saying the drug is BCS class I, so no B study is required. That is not an acceptable scientific conclusion by itself. BCS classification is only the beginning of the assessment. You must evaluate the complete eligibility criteria. What does high solubility actually mean? We keep using the term high solubility, but what exactly does it mean? In the BCS context, high solubility is not simply based on whether somebody describes a drug as soluble or freely soluble in a pharmacopoeial monograph. The assessment is based on defined scientific criteria. In general, the highest single therapeutic dose, or the relevant dose criterion specified in the applicable guideline, is evaluated to determine whether it can dissolve in a defined volume of aqueous media across the relevant physiological pH range. You may commonly see a volume of 250 milliliters discussed in BCS assessments. Conceptually, this represents approximately 1 glass of water, similar to the volume that may be used when administering an oral dosage form. The solubility assessment is performed across a relevant pH range because the gastrointestinal tract does not have one constant pH. The stomach has an acidic environment. The pH changes as we move into the intestine, and the solubility of many drug substances is pH dependent. A drug may dissolve very well at acidic pH but poorly at a higher pH, or the opposite may happen. Therefore, BCS solubility classification requires proper scientific data. It should not be based on assumption. Now let us understand permeability. Permeability tells us how effectively the drug is absorbed across the intestinal membrane. In regulatory science, permeability classification may be supported using accepted human data or other scientifically justified approaches permitted by the applicable guideline. The basic question is what proportion of the administered drug is absorbed. For a highly permeable drug, the extent of absorption is high. Again, the exact criteria and acceptable methods must be checked against the relevant regulatory guideline. And this is important because permeability is not something that you can conclude simply from a routine QC test. You cannot perform assay dissolution and disintegration testing and then declare, yes, this drug has high permeability. Permeability classification requires appropriate scientific evidence. Now let us come to the practical part. If we are trying to avoid an in vivo bioequivalence study, how do we compare the test and reference products? One of the most important tools is comparative dissolution testing. Let us understand the basic logic. We take the test product, we take the reference product, and we evaluate how the drug is released into dissolution media over time. But we do not simply test one tablet from each product and compare the final percentage dissolved. A proper comparative dissolution assessment involves defined test conditions. Multiple dosage units are tested, samples are collected at appropriate time points, and dissolution profiles are generated. For example, you may have dissolution results at 5 minutes, 10 minutes, 15 minutes, 20 minutes, 30 minutes, and later time points, depending on the study design and applicable guidance. When you plot percentage drug dissolved against time, you obtain a dissolution profile. Now imagine the reference product reaches 85% dissolution very rapidly. The test product also reaches a similar level at approximately the same stage. Their profiles may appear comparable, but now imagine another situation. The reference product reaches 85% dissolution at 15 minutes. The test product reaches only 45% at 15 minutes and requires 45 minutes to reach a similar level. Even if both products eventually reach 90 or 95% dissolution, their release behavior is clearly different. This is why looking only at the final dissolution result can be misleading. Comparative dissolution is about the profile, not simply the final number. What is the F2 similarity factor? At this stage, you may have heard about something called the F2 similarity factor. The F2 value is a mathematical tool used to compare two dissolution profiles under applicable conditions. I am not going to turn this episode into a mathematics class, but conceptually, the purpose is very simple. We want to know how similar is the dissolution profile of the test product to the dissolution profile of the reference product. An F2 value within the generally accepted similarity range can support the conclusion that the two dissolution profiles are similar, subject to the conditions of the applicable guideline. You will commonly hear that an F2 value between 50 and 100 indicates similarity. But please understand that calculation and interpretation of F2 have specific requirements. The number of units tested matters. The number and selection of time points matter. Variability matters, and there are conditions regarding the data used in the calculation. Therefore, please do not simply put dissolution values into an online F2 calculator and conclude that your product qualifies for a biowaiver. Regulatory science does not work like that. The complete study design must be scientifically and regulatorily appropriate. In certain situations where both products dissolve very rapidly, the applicable guidance may not require an F2 calculation. Again, this depends on the specific regulatory criteria. Why are multiple dissolution media used? Now, another important question. Why don't we simply compare dissolution using the routine QC dissolution method? Because the objective is different. A routine QC dissolution test is designed to assess product quality against an approved specification. A biowaiver dissolution assessment is intended to compare the behavior of the test and reference products under physiologically relevant conditions defined by the applicable guidance. Therefore, comparative dissolution studies for BCS-based biowaiver assessment are generally performed across multiple pH conditions. You may commonly see media representing approximately pH 1.2, pH 4.5, and pH 6.8. Why? Because the dosage form moves through different gastrointestinal environments. A product may show similar dissolution to the reference in acidic conditions, but very different behavior at pH 6.8. If we tested only one medium, we might completely miss this difference. This is why multimedia dissolution profiling is so important. We are trying to understand whether the test and reference products behave similarly across relevant gastrointestinal pH conditions. A simple practical example. Let us take a hypothetical example. Suppose company A is developing a generic immediate release tablet. The drug substance is scientifically established as belonging to an eligible BCS category. The company evaluates the reference product. Then it develops the generic formulation. The test product and reference product are subjected to comparative dissolution testing. At pH 1.2, both products dissolve rapidly. At pH 4.5, both products show comparable dissolution profiles. At pH 6.8, both products again demonstrate appropriate similarity. The formulation meets the relevant excipient requirements. The drug substance meets the applicable solubility and permeability criteria. The dosage form is eligible, and all other regulatory conditions are satisfied. In such a situation, the company may submit a scientifically justified request for a BCS-based biowaiver instead of conducting an in vivo BE study. Now change just one part of the example. At pH 6.8, the reference product dissolves rapidly, but the test product shows significantly slower dissolution. Now we have a concern. Can we confidently predict that the two products will behave similarly in the human body? Maybe not. The biowaiver justification may no longer be adequate. The company may need to reformulate the product, generate additional scientific evidence, or conduct an in vivo bioequivalence study. This is why dissolution development during generic formulation is not simply a QC activity. It can directly influence the regulatory development strategy. A biowaiver is not a shortcut. This is one point I want every student and pharma professional to understand. A biowaiver is not a shortcut. It is a science-based regulatory pathway. Sometimes people hear the word waiver and think no clinical study, so easy development. No. In fact, preparing a scientifically strong biowaiver package requires a deep understanding of the drug substance and drug product. You need reliable solubility information. You need appropriate permeability evidence. You need detailed formulation information. You need comparative dissolution data. You need analytical methods capable of generating reliable results. You need appropriate reference product selection. And you need a scientifically sound regulatory justification. If the scientific evidence is weak, the regulator can ask for an in vivo bioequivalence study. So the objective of a biowaiver is not to escape science. The objective is to use science more efficiently. Biowaiver versus in vivo B study. Let us compare the two approaches conceptually. In an in vivo B study, we directly compare drug exposure in human subjects. We administer the test and reference products, we collect blood samples, and we compare pharmacokinetic parameters. In a BCS-based biowaiver, we use knowledge of the drug's solubility and permeability, combined with formulation assessment and comparative dissolution behavior to scientifically predict that an in vivo difference is unlikely to be clinically meaningful. One approach directly measures human pharmacokinetics. The other uses a validated scientific framework to determine whether the human study may be unnecessary. This is why BCS is so powerful. It connects pharmaceutical science, biopharmaceutics, dissolution, and regulatory decision making. Are biowaivers only BCS based? Now, here is another important point. When people hear the word biowaiver, they often think only about BCS-based biowaivers. But the broader concept of waiving an in vivo B requirement can arise in other regulatory situations as well. For example, under appropriate conditions, additional strengths of a pharmaceutical product may not always require separate in vivo bioequivalence studies. Imagine a company developing three strengths of an immediate release tablet: 10 milligrams, 20 milligrams, and 40 milligrams. Does the company always need to conduct three completely separate in vivo B studies? Not necessarily. Depending on the formulation proportionality, manufacturing process, pharmacokinetic characteristics, dissolution behavior, and applicable regulatory requirements, an in vivo study may be conducted on one strength and a waiver may be requested for other strengths. This is often referred to as an additional strength biowaiver or a waiver for other strengths. But again, the waiver is not automatic. The company must demonstrate that the scientific conditions are satisfied. We will discuss this concept in much more detail in a future episode because additional strength biowaivers are extremely important in generic product development. Products that are generally not simple, BCS biowaiver candidates. Now let us also understand where we need to be cautious. BCS-based biowaiver principles are primarily associated with eligible immediate release oral solid dosage forms intended for systemic action, subject to regulatory criteria. Modified release products are a different situation. Enteric coated products require specific consideration. Narrow therapeutic index drugs may have additional restrictions or may not be accepted under certain biowaiver pathways. Products where excipients can significantly influence absorption require careful assessment, and drugs with complex absorption behavior may not be suitable for a simple BCS-based approach. Therefore, you cannot take the BCS concept and apply it blindly to every oral dosage form. Always ask four questions. What is the drug substance? What is the dosage form? What is the formulation? And what does the applicable regulatory guideline permit? These four questions can prevent many regulatory mistakes. Why biowaivers matter to the pharmaceutical industry? Now let us look at the industry perspective. Why are biowaivers important? First, they can reduce unnecessary exposure of human subjects to clinical studies. If strong scientific evidence can reliably demonstrate equivalence, conducting an additional human study may not always provide meaningful additional information. Second, biowaivers can reduce development timelines. An in vivo BE study requires protocol development, regulatory and ethics approvals as applicable, clinical site activities, subject recruitment, dosing, bioanalysis, pharmacokinetic analysis, and final reporting. A scientifically justified biowaiver pathway may reduce some of these activities. Third, biowaivers can reduce development costs. Bioequivalence studies can be a significant part of generic product development expenditure. And fourth, biowaivers encourage companies to understand their products scientifically. Solubility, permeability, dissolution, excipient functionality, formulation behavior. These are not simply academic concepts, they can directly influence regulatory strategy. A message for pharma students. If you are a pharmacy student watching this series, you may be thinking, Sir, do I really need to know BCS classification for a pharma job? My answer is yes, especially if you want to work in formulation development, regulatory affairs, bioequivalence, clinical research, or even certain areas of quality and analytical development. During an interview, somebody may ask you, what is a BCS Class II drug? Do not simply answer low solubility and high permeability. That is correct, but add one more line. Say, because solubility is low, dissolution may become a rate-limiting factor for absorption, and formulation differences can significantly influence bioavailability. Now your answer shows understanding, not memorization. Similarly, if somebody asks, why are BCS Class I drugs suitable candidates for a biowaiver? You can say, because they have high solubility and high permeability, and when eligible, immediate release test and reference products show appropriate rapid and similar dissolution, their in vivo performance may be sufficiently predictable under the BCS framework. That is a much stronger interview answer. Let us revise the complete concept. Before we close today's episode, let us quickly connect everything. A biowaiver is a regulatory pathway under which an in vivo bioequivalence study may be waived when sufficient scientific evidence is available. One important approach is the BCS-based biowaiver. BCS classifies drug substances mainly on the basis of solubility and permeability. Class I drugs have high solubility and high permeability. Class II drugs have low solubility and high permeability. Class III drugs have high solubility and low permeability, and Class IV drugs have low solubility and low permeability. Eligible Class I and under defined conditions, Class III immediate release products may be considered for BCS-based biowaiver approaches under applicable regulatory frameworks. Comparative dissolution testing plays a major role. The test and reference products are evaluated under defined conditions and across relevant dissolution media. Dissolution profiles are compared. The F2 similarity factor may be used where applicable. Formulation composition, particularly excipients, may also be extremely important. And most importantly, a biowaiver is not automatic. It must be scientifically justified and acceptable under the applicable regulatory requirements. So the next time somebody tells you a biowaiver means you don't have to do a BE study, I want you to add one important sentence. Yes, but only when science gives us sufficient confidence that the in vivo study can be waived. That is the real concept of a biowaiver. In the next episode of our Bioequivalence Studies Masterclass, we will go one step deeper into practical BE study design and understand an extremely important concept: crossover study design. Why does the same volunteer receive both the test and reference products? Why are there different study periods? What is a sequence? What is a washout period? And why is the two-period, two-sequence crossover design so commonly used in bioequivalence studies? We will understand all of this using a simple practical example. If you are following this bioequivalence masterclass, make sure you watch the episodes in sequence because every episode is connected to the next. If you found this explanation useful, please like this video and share it with pharmacy students and pharmaceutical professionals. And if you have any question about biowaivers, BCS classification, or comparative dissolution studies, write your question in the comments. I personally read your comments, and your questions also help me decide what concepts need more explanation in future episodes. Subscribe to PharmaHealth Insights and continue learning with me. Thank you for watching, and I will see you in the next episode of the Bioequivalence Studies Masterclass.